Welcome everyone to week number 3 in Biology 120. This week is all about the classification of life and how we group things together in order to identify them and understand them, and cells and also viruses. Especially in lab, we have a little bit about viruses. So let's take a look at what is happening. So last week, you learned about the chemistry of life, the importance of water, all the different properties of water, the fact that it's a solvent, that it absorbs heat, that it adheres and it coheres, and it has so many functions in cells and in bodies and organisms, and it is absolutely essential for life. We learned about the element carbon and the fact that it forms 4 bonds with other atoms or molecules and can make very large complex structures that we call macromolecules. And we learned about the 4 groups of organic molecules: carbohydrates, lipids, proteins, and nucleic acids, and the importance of them. And you did answer that question about whether nucleic acids are found on food labels, and the answer is no. And the reason is because it's made up basically of a sugar and part of an amino acid, which is protein. So you don't have to put nucleic acids on a food label. The topic for this week is classification. And so classification is putting things in groups, right? And we naturally do this as human beings, but lots of other animals do that too. For example, my old dog, Charlie, he hated squirrels. He loved cats. He loved every other kind of animal, all people. But the one thing he hated was a squirrel. And when he saw a new squirrel, he would take off running after it if he could and bark and bark and bark and bark. And he did that with nothing else on the planet. He classified squirrels as squirrels, and when he saw a new squirrel, he knew exactly what it was. Well, the whole point of this is that we naturally do this as human beings, and so, of course, we do this as scientists too. Well, why do we do this? We do this so we can predict. If we figure out how this thing fits into a group, then we can predict what's going to happen with that thing. And that's the advantage to classifying. So we do this, and we do this with organisms. In fact, that we classify into 3 major domains. We have the domain Eukarya, we have the domain Eubacteria, and then we have the domain Archaea. So Archaea contains all the archaebacteria, which live in very extreme environments. So we rarely would come in contact with archaebacteria, but they're out there in the environment, and they have been on Earth for billions of years. Then we have the domain Eubacteria that contains other bacteria that we normally would come in contact with. Those out in the soil, those on our skin and our digestive tract, in water, just everywhere. There's so many bacteria in so many different environments. In your septic tank, lots and lots of places. So eubacteria means true bacteria. So far the 2 domains contain bacteria, and then the 3rd domain, Eukarya, contains all the other organisms. Eukarya means true nucleus. So these particular organisms have more complex cells than bacteria do. And so what does this include? All the plants, all the animals, we're animals, all the fungi, okay, a shelf fungus, a mushroom, a mold, yeast, those are fungi, and then the protist, and that's everything else that is eukaryotic but doesn't fit into the other categories: plant, animal, or fungus. And so those are the 4 groups or kingdoms really in the domain Eukarya. So we classify. And it's important to understand the difference between eukaryotic cells and cell structures, that's our cells, and bacterial cell structures, which are much more simple, no organized organelles inside the cells, no nucleus, mitochondria, etc. There are major differences in our cell structures, and this is how antibiotics work. Because if you take an antibiotic, which is a chemical that kills cells, If you take an antibiotic, you want to kill the bacterial cells and not kill your cells. And so there have to be differences in our cell structures in order for antibiotics to work. And you're going to discuss that. Read your chapters in your OpenStax textbook. Remember, it is free. Go online, just at least skim through it. Chapters 20, section 1 and 4, and chapter 4. So be sure and do that so that you can get the background information for this week. Read the Module 3 overview and watch the videos. The videos are very important. I also will have an additional video in this announcement. The Harvard Medical School experiment on antibiotic resistance in bacteria. They made a huge plate of agar inoculated with E. coli and watch what happened to the E. coli at different antibiotic concentrations over an 11-day period. And this really is one of the most amazing studies I have ever seen in my life. They use time-lapse photography to show you what happened with the E. coli and how fast they become resistant to that antibiotic. It is absolutely amazing. And it shows you also what can happen in your body. People say, it takes a long time for resistance to occur. Oh, no. Watch how fast it happens and to what degree the E. coli are antibiotic resistant. It is just amazing. So please do watch that because it is phenomenal. All right, what do you have to do this week? The discussion board postings. So you have discussion board postings and final project. This week, you are going to discuss why antibiotics target bacterial cells and not ours. So you have to know some differences between the two types of cells and what antibiotics actually target in bacterial cells. So that's important, and you need to look that up. And that's found in your textbook, so go back and look at it. Also, you're going to talk about how our use of antibiotics increases the rate of antibiotic resistance. Antibiotic resistance is going to happen. Bacteria figure out ways to survive, all right, no matter what the stressor is. They figure it out because they can mutate their own DNA, and they have all different ways to do this. But why is the particular use of antibiotics increasing the rate of antibiotic resistance? Do we take too many? Do we take them incorrectly? Take a look at that because there's lots of reasons that this happens. And this includes the production of superbugs like MRSA, methicillin-resistant Staphylococcus aureus, which I always was particularly interested in. I did a study when I was at York Technical College for 17 years to find out how many people carry Staph aureus, just a particular type of bacteria that causes staph infections. All of y'all have heard of that. And then after a while, when methicillin resistance popped up in Staph aureus, I started looking at, well, how many of those students who were positive for Staph aureus also carried MRSA? And there were a number. And a lot of my students had worked in healthcare, so I wanted to know if they were picking this up from the healthcare setting. And the answer to that is no, that they were actually coming in contact with MRSA out in the community instead of getting it in a healthcare setting. So that was interesting. So you're going to discuss that. In your response posts, you are going to have a bit of a discussion about antibiotic use in livestock. So antibiotics have been given to poultry and beef cows and dairy cows and pigs since the 1950s. And the reason, hey, there just went a car by. The reason that they've been given antibiotics is because they grow bigger, faster, or they lay eggs bigger and faster because they have antibiotics in their feed. They are not being given antibiotics because the animals are sick. You need to realize that. But they increase production in livestock. The antibiotic use does. Why does that happen? Because the antibiotics take care of what are called silent infections. All of us, human beings included, have silent infections, where our bodies are fighting off some kind of infection right as of this moment. Okay? But we're being very successful at it. The thing is that it takes energy to do that. Some of our resources, some of our energy has to go to fighting off silent infections. Well, if you can cure all of those, then all of your energy can go into meat production or egg laying or whatever. So that's why antibiotic use has happened in livestock just in their regular feed for decades now. And we are getting away from that. So you're going to discuss that topic. Okay, then you're going to begin your final project. So hopefully you have looked at that list and you have chosen a topic. If you want to do anything different, you have to email me and get an okay before you do it, because you don't really understand yet how much time and effort and how much information you're going to need for this final project. If you tell me what it is, then I can be the judge of that. So I will help you figure out what to do. Take a look at that milestone 1 guidelines and rubric, because that tells you how to put this together. It tells you what to include. This is extremely important, okay? And use the correct format for this milestone 1. It is an outline with details. It is not in essay form, not like your journals, okay? And label your sections, and I'm going to talk about that again in a second. Okay, this is what the milestone 1 guidelines and rubric looks like. There's your list of topics again. You're going to make this outline that has detail in it. So it's like bullet points, but you have information under each bullet, okay? You're going to use this to make your final presentation in slide form, like in PowerPoint, okay? So keep that in mind. Each section is going to be a slide. Just realize that. So there's your list of topics, everything from population ecology to water pollution to deforestation to climate change, etc. There's lots of choices. If you don't like those or you want to do something else, remember you must email me and get it okayed. So for milestone 1, you're doing the background information for this particular topic. You're going to introduce the topic, so explain what it is. Don't you think that's pretty important at the beginning of a presentation? If you say genetic modification, some people in your audience aren't going to know what that is, so explain it, describe it. That does not mean 2 words. That means a paragraph. Also, tell why is this significant to you or interesting to you, one or the other. Why is it significant or interesting in the field of biology? All right, and support your response with real life examples. So if you're talking about genetic modification, you might include the fact that golden rice has been produced for decades now to help with the world's feeding the world's population, because it is very nutritious rice. Then you have a section on topics. So notice that there's an outline format in the instructions here. Your outline should look like this. Big numeral 1, introduction, and a couple of paragraphs under there. Big numeral 2, topic. Then A, where. Where does this happen? If you're talking about global warming and climate change, well, it happens throughout the whole biosphere. So you're going to explain what that is. If you're talking about, for example, the pollution in a river near you, near where you live, then you're going to describe that particular aquatic ecosystem of that river. All right, so you see where this is going? If you're talking about, let me look at one of the others, endangered species, like maybe you've chosen bald eagles or something of the sort. Where are they located? Are they located along the coast? Are they in forest? Are they along marshes? Of course, there are lots in Alaska. So where particularly do you find bald eagles, and where is their natural habitat? How does this process happen? If you're talking about eutrophication, how does all this mineral, excuse me just a second, how does all this mineral and nutrient buildup happen in a body of water? Where's it coming from? How does it occur? Of course, a lot of this is sediment, so you have to discuss that. Plus, there's a turnover in bodies of water where the temperature changes and those sediment, the nutrients in the sediment are brought to the surface. So you need to explain that. Which species are affected? So are there bacteria that are affected? Are there species of fish that are affected? Are there human beings that are affected? Are all species affected? You need to describe these. Also, you need to talk about who benefits from this particular process. If you're talking about the pollution of that river near your house, then which industries are benefiting from this? Which ones are dumping waste products in? Or are homes allowing sewage from septic tanks to flow into the river? Or waste from hog farms? Are they being dumped into the river that changes this ecosystem? So you need to talk about who benefits. Or it might be, if you're talking about red tides in Florida, then it could be the algae or the different produce that are benefiting from this particular phenomenon. Okay, so that's the basics of what you need to do. And so this is a good bit of research. Use the Shapiro Library. Do not just Google everything, because you're going to come up with some crazy references, I'm telling you. There are too many opinion pieces out there that aren't based on anything besides someone's opinion, and that is not what we want to include. All right, your tip of the week is to put your assignments in the correct format. What have I just been talking about with the final project, milestone 1? You have an outline format, Word document, okay? So here I have the guidelines. I took out the information from that milestone 1. Create an outline. But the outline has to have detail in it. It's not just a word. No, that's not going to help you. This outline is going to be used to make slides for your final project. So remember that. How should this be submitted? As a Word document, double spaced. I don't really care about the font, but don't make it crazy. And don't make it pink colored or yellow where I can't read it, right? I wear glasses for a reason. I don't want it to be hard to read, right? So make it easy for me to read. 1-inch margins. I do not measure any of this. I mean, that would be crazy. Just make it easy to read. That's all I'm asking. And especially make it double spaced, because that makes it easy to read too. Maximum of about 2 pages. That sounds about right, not including the references. You will use at least 3 references for every milestone. You have 3 milestones, okay? Provide in-text citations. So if you get information from somewhere, you have to cite that in the text. All right, and there's a specific way to do that, APA format. And you'll also have a reference section, okay, with every one of these milestones, because you have to tell me what your references are so I know what they are. Okay, I think that's about it. But just realize that every assignment has a particular format, and that's out there in the world too. If your boss tells you you need to make a presentation that's about 30 minutes long, needs to be PowerPoint, you have to follow the instructions. So make sure you use the right format. And you're going to see that this changes in the final project milestones. So just realize that. This first one, outline format, Word document. Okay, I hope that you have a wonderful week. And as always, let me know if you need anything. I'm here for you. I'll be grading sooner this week because I'm back from being out of town. So I'll get to grading faster this week, which is awesome. So I hope that you have a wonderful week, and let me know if you need anything.